That was wonderful. Thank you so much for that presentation, Ophelia. Yeah, we have a few questions here for you. Let's see, I think, yeah, I'll just go in the order of the votes. First question is, do you think hydrogen sulfide production in the human gut induces metal starvation more generally across the ecosystem? Is there any evidence for this effect?
Yeah, that's a great question. I haven't looked into the literature, but I really should. Hydrogen sulfide is produced by gut bacteria. It's also produced by the host. So the contribution of hydrogen sulfide produced by gut bacteria relative to the host, I'm not quite sure if it's what percentage that is, but I know the host has a substantial contribution as well. I would imagine that it could sequester different metals. But yeah, I don't know. That's a great question. I'll have to look into that more and see what's known. Yeah.
And then related to that, I think you mentioned this in your talk. I think I might have missed it, but when you were talking about the mechanism of the ablation of the metromidazole effect, was that due to the sequestration of the metals, you think?
Sorry, I missed the beginning.
When C. diff was growing in the presence of deep Piger, you see the antibiotic becomes ineffective. Do you think that's because of the hydrogen sulfide sequestering like the metals?
Yes, so we did an experiment where we take media, conditioned media by deep Piger, and we actually add back the metals, and then C. difficile becomes sensitive to the antibiotic again. So it does seem that we can rescue this effect with metal supplementation. So yeah, so we do think that might be a major mechanism. So yeah, so I think that's our current working hypothesis.
And then another one that's sort of related to that, it was kind of fascinating, like when you show the like those MIC plots with the C. diff and D. Piger, and there was this like sort of interesting phenomena where it looks like in the presence of D. Piger, C. diff actually grows better with higher concentrations of antibiotic.
Yeah, that's a good question. I think
strange. Why is that, you think?
Yeah, that's a good question. I don't know. We would have to kind of look at the cell size also under the microscope, which we did in some experiments. I don't know if we did in that one, but like when we look at OD, it is measuring also how big the cells are. So it's a bulk measurement of the solution. It's not really telling us like how many cells are there. And yeah, so I don't know if that is reflected in like the number of cells that are changing or whether it's like a cell size thing. Yeah, that's another good question. I don't know if we investigated that in depth. And so anyway, that's something else that we could look into.
That's a really good point. Yeah, that maybe they're just getting like longer or wider.
Yes, yeah, it is possible.
Okay, let's see, a couple more questions for you. Yeah, you mentioned the power of using models like GLV and neural networks, and you demonstrated in your in vitro communities how you can like use those. The question is, how applicable do you think these are to in vivo gut microbiomes?
It's very hard. In vivo experiments are extremely low throughput. So when we do mouse experiments, we just have a limited set of conditions. We do get time series, but the time series is not very rich in information unless there's perturbations. So usually the community will just converge to some steady state and just stay there, so it's not particularly informative. So anyway, we haven't modeled in vivo data in mice. I think that there is potential there for thinking about what kinds of experiments we could do to build informative models, and people have built models with in vivo data. It's just how much we actually trust the parameters of the model. So you certainly can do it. It's just, do we trust the parameters?
Yeah.
And it's also hard to kind of validate that. I mean, you could design experiments to test it, but yeah, it's a pretty involved effort. But with such limited data, it becomes challenging. So I do think that models that are more mechanistic, that have less flexibility, maybe it would be, they would have more constraints, and there wouldn't be as like, you wouldn't need as much data for parameterizing those models. The limitation is that we don't necessarily know those models are representative of what's happening in vivo. So I think this is an interesting area that we'll have to think a lot harder about. What we're doing these days is thinking about how to build models with in vitro or ex vivo systems where we have, co-cultures of like a mini gut model, which is higher throughput, and then use that to predict what happens in vivo, like design communities that are interesting and test in vivo, and then we test those in vivo. But we don't use the in vivo data as part of our modeling, which is limiting, So I think that is a future direction that I need to spend some time thinking a lot more about, but it's just very challenging when you have such a limited number of conditions, especially when you're thinking about like very data hungry models like, machine learning. But maybe with human data, but then there's so much variability, it's like, we're exploring some things with a human data set. So we'll see where that goes, but it's challenging.
It'll be
and it's important. Yeah,
it'll be interesting to see how that develops, because I know that is sort of a controversial area where some people claim it can be done, and other people have argued that like you were saying, the dynamic, you're not really seeing dynamics, you're seeing steady states.
In mice, yes. I think when you have perturbations, though, you could get some more rich dynamics. But it's, I think anyone can build a model with any data. The question is it a good model, right? I think that's the challenge, is like, we can all go and build models with data, but if the parameters are not constrained, they might not necessarily represent what's happening in reality. So it's,
Yes. It's like that saying, all models are wrong, but some models are useful.
Right, exactly.